All right, hi everyone. Welcome back to our second day of the Ensemble Browser Workshop. My name is Louise, and I'm here with Ben today. And for today, I'll be delivering all of the modules. Um, and then tomorrow, Ben will be delivering a couple more modules as well. Um, so just to go back to our schedule. So yesterday, Ben introduced you to Ensemble and how to view genes and transcripts on the browser. And today I'll be talking about variation and regulation data available in Ensemble. Okay, so let's get started. Um, so first up, we'll be talking about variation data in Ensemble. Um, Sorry, my computer just froze. Okay, I hope you can see this. If you can't, let me know. And if you want me to speak up or if I'm talking a bit too loudly, just put it down in the chat. Okay, so in this module, I'll talk about how variants are classified, the species for which variation data is available, and the sources from which Ensemble retrieves data from. So we will explore the variation data in Ensemble during the demonstration, and in the end, you will have some time for some exercises as well. Okay, let's talk about variation types. So we can group variants according to their size or the number of nucleotides that are affected. Variations can come in a small scale where only one or a few nucleotides are of a gene are affected. So for example, here we have two sequences. The top sequence is the reference sequence. The bottom sequence is our own sequence. And small scale variation types include small insertions. So for example, here, or deletions, which are also called dips or indels. Or these can also be single nucleotide polymorphisms or SNPs. So these are changes in the nucleotide. In this example, the reference sequence has the A variant or A allele, and our sequence has the T allele or T variant. Variations can also come in a large scale, such as in the chromosomal structure, and these are also called structural variations. The types of structural variation are copy number variations, or CNVs. So, for example, when you have a genomic sequence with a lot of repetitive regions, and they can also come in large deletions, for example, where a large region in the chromosome is deleted. And they can also come in duplications, so where a region in the chromosome is duplicated, also insertions and translocations. Variation data is available for a number of different species on the Ensembl browser. So the Ensembl browser is dedicated for vertebrates, but it's also available in the Ensembl genomes browser, so which is dedicated for non-vertebrate species. Um, and there is no variation data available for bacteria as there are too many of them, and there's a lot of recombination in the bacterial genomes. I'll show you how you can find out if we have variation data available for the species that you are interested in. So where does the variation data come from? In general, there are 4 steps in the Ensembl variation data process. So first, the variation data is imported from publicly available databases. During the import, the data is being checked for the quality, and then we compare the data that passed the quality control with other databases to link them with associated data or studies. And finally, we analyze the data and process them to make them available in Ensembl. So let's look at each step in more detail. In the first step, we import variation data from publicly available archives and sources. We search variation data from a number of different databases. So for non-vertebrates, we import variation data from the European Nucleotide Archive, ENA. Oh, sorry. And also the European Variations Archive, EVA. And also some species-specific projects and collaborations. For human, for example, we retrieve data from dbSNP for short of genetic variations. We retrieve structural variation data from the database of genomic variants, so DGVA, DGV archive, sorry. And for somatic variation data, we retrieve these from Cosmic, and variants implicated in cancer from the cancer gene census. If you're interested in finding out more about the individual sources that we retrieve data from, you can click this link here on the presentation. In the second step of the Ensembl variation process, we apply some quality control measures on the data that we imported. Here we map the variants to a reference assembly. We compare the reported alleles with the reference genome, and we exclude suspect variants. So for example, alleles that contain ambiguity codes or non-nucleotide characters. And again, for more information, because it's quite, this is only an overview that I'm giving you, but if you're interested in the individual steps in more detail, you can find out more in our documentation pages. In the third step, we link the variation data that pass the quality control to other databases. So here we add value to the data, for example, by linking a variant to allele frequencies, or phenotype, or any publications. Excuse me. Allele frequencies are one of the main types of data that we link to our imported variants. Allele frequencies describe how often an allele is present in a population. So for this, we link imported data with databases such as for human, for example, the 1000 Genomes Project, NOMAD, UK10K, and TopMed. We also link imported variation data with phenotype and disease data from a range of different sources, depending on the species. So for example, for human, we link variation data to the Human Gene Mutation Database, or HGMD, which you can see here, and ClinVar. For mouse, we link variation data to the International Mouse Phenotyping Consortium, or IMPC. And we retrieve information such as the clinical significance of the variant, inheritance type, the reported gene and risk allele. And for genome-wide association studies, or GWAS, we also retrieve statistical information, for example, p-values, odds ratio, and beta coefficient. In the final step, we apply our own analyses on the variation data that we imported and that pass the quality control. So here we predict the variant consequence, the protein function, and we calculate linkage disequilibrium and variant conservation across different species. So how do we predict the variation consequences? In general, we take each variant and plot these along the genome. And we predict the functional consequence of the variant depending on where the variant falls on a gene. So for example, if a variant were to fall outside the gene, such as the 5' or 3' region, the variant is unlikely to affect the gene itself, but likely to affect the regulation. So for example, the mRNA transcription and transcription activity. If a variant were to fall within the gene, but not within a coding sequence, so the yellow boxes here represent coding sequences, coding regions, sorry. The variant is unlikely. So if it were to fall in the non-coding regions, the variant, it would be unlikely to affect the gene itself, but might affect the mRNA stability. So for example, splicing or translation efficiency. If a variant were to fall within the coding regions, so the yellow boxes here in this image, these can have an effect on the gene in two ways. So these can have a synonymous or a non-synonymous effect. And for synonymous variations, the protein sequence remains the same. Therefore, there is no change in the amino acid. And for non-synonymous or missense variations, there is a change in the amino acid and therefore the protein sequence. So it's likely that this type of variant would have a greater consequence to the gene itself and also the function of the protein. So to summarize, in Ensembl, we plot each variant to the genome, and depending on where the variant falls within a gene, we assign a consequence to the variant. And these variant consequences have special terms. So we also call these sequence ontology, or SO, and it is a standardized way of describing sequence-based features. Just like how we have a standardized way of describing gene function. So yesterday, Ben talked about gene ontology, or GO, GO terms. In this case, each SO or SO term is color-coded, and you can read more about sequence ontology in the link here. And also, you won't have to memorize any of these colors because they will be available to you on the Ensembl browser. So I'll show you where you can find a legend for each color. When we talk about non-synonymous or missense variants, we rank them by their pathogenicity. So there are different algorithms that rank these changes in the amino acid sequence based on how conserved a protein is, and also how if there is a chemical change in the amino acid position and the 3D structure of the protein. But please note that these are predictions and not facts. So it is always good practice to validate your predictions experimentally. Ensembl uses a number of different pathogenicity tools which rank these missense variants according to a scoring system. So for example, the SIFT score, the first one here, ranges from 0 to 1, with 1 predicting a deleterious effect, with 0 predicting a deleterious effect. For polyphen 2, oops, sorry, my mouse is playing up. So we've done Swift. For polyphen 2, the score is a bit different, so the scoring system is inverted. A 1 indicates a damaging effect of the variant, and a 0 indicates a benign effect. Another scoring system, the CAD score, ranges from 100 to 0, with 0 indicating a likely benign effect. And then a couple more of these pathogenicity scores, so REVEL, Meta-LR, and Mutation Assessor. Um, their scoring systems range from 0 to 1, with 1 indicating a damaging effect on the variant. Finally, I want to talk about reference alleles in variation data. If you remember from yesterday, Ben explained that genome assembly is made up of chunks of sequences from individuals, like a jigsaw puzzle. So when you take a sequence in the human genome assembly and look at a particular variant, for example, T here, uh, it does not necessarily mean that it is a representation of all the human individuals around the world. So this T allele is found in this particular person. However, it does not mean that the majority of people have this T allele. So she might actually be a person who carries an allele which could cause susceptibility in a disease. So when we talk about reference alleles, it's not always the major allele or the allele that is found most frequently in a population. But most of the time, the reference allele is the major allele. But just keep in mind that this might not be the case. Okay, so for our first demonstration, we're going to look at the MCM6 gene in human, and we're going to find variants in the gene, variants in the gene region, and then also I'll show you how to look up a particular variant on Ensembl. But before I begin with the demonstration, I just want to show you how you can find out if there is variation data available for the species that you are interested in. So I'll open up a new tab, and the first thing I'm going to do is show you if there's variation data available for your species. So I'll go to ensemblegenomes.org, and for example, I'll go to the so this will be the same for any of the Ensembl sites, but I'll use Ensembl plants as an example. Excuse me. So I click go to Ensembl plants, and then once you're on the home page, I'm just gonna zoom in a little bit so it's a bit easier for you to see. So this is the home page, and then here on the left-hand side, you'll see a link that says View Full List of All Species. So if you click that, Ben has shown you this exact same table, I think, on the Ensembl browser, but the structure is all the same. So to find out if there is actually variation data available for your species, you just need to check this column here on the right-hand side. So I'm just going to click it so that it sorts the table according to the availability of variation data. And if you have a check in this column, it means that there is variation data available for this particular species, so for Arabidopsis here. And this is also how you can find out if there's regulation data available. So I'll talk about regulation more later. But again, if there is a tick in this column, it means there is variation or regulation data available. And you can do the same for any of the other Ensembl sites, so the protists and fungi and so on. Okay, so let me actually go to the Ensembl site so I can start with a demonstration. So in the address bar, I will type in ensemble.org. Let's zoom in again. And this takes you to the homepage, which you have explored yesterday already. So we are interested in the MCM6 gene in human, and we want to find out the variants within the gene and also within the region. So I will, mm, ah, sorry, I was looking for the chat window. So I will paste the gene name into the chat box so you can follow along if you like, or you can just watch me do the demonstration on your screen. And what we'll do is we'll search for the gene first. So we're interested in humans. So here in the middle of the page, there is a drop-down menu and drop-down option. And when you click it, you'll select human because we're interested in human. And then underneath in the search bar, I will type in, or I will paste the gene name, so MCM6. And then I'll click go on the right-hand side. Okay, so this will be our first hit, MCM6 human gene. So I'll click the first link, which takes us to the gene tab. So you can see which tab you're in by looking here at the top of the page, just underneath the species name. Okay, so first I want to show you how you can find all the different variants found within this gene. So if you look on the left-hand side here in this panel, there is a section called genetic variation a bit further down. If you click the variant table, it opens, it refreshes the page on the right-hand side, and this actually gives us a table of all the different variants found within this gene. So it might take a bit of time to load, but it actually loaded quite quickly for me. So here we have this table. The headers look a bit funny on my screen. That's because I'm zoomed in. But if you are doing it on your own laptop, or if you zoom out, you can see that it actually adjusts. So just bear with me for these zoomed in version. Okay, so we have this table here with a lot of different information on the variant. So we've got the variant ID, we've got the position, so the chromosome number, and then the base pair position. We've got the different alleles. So the first allele is always the reference allele. The second allele is the alternative allele. Then we've got global minor allele frequency if it's available. If you're not sure what a column represents, you can always hover over the headers. So whenever there is a dotted, if there is a dotted underline, it means that there is an explanation or a description available for you. Then we also have got the class, so the type of variant. In this case, for this first one, it's the SNP. Then the source, so which database this is coming from, the different evidences that are available. So this was, for example, studied in the NOMAD project. Then we also have the clinical significance, if available, the consequence type, then the amino acid change, if it does change. So in this case, it's glutamic acid to glycine, and also the amino acid coordinates. And then finally, at the end of the table, we've got all the different pathogenicity scores that are available. So so don't forget these scores are just predictions, and therefore they can contradict each other. So it's always best practice to validate your predictions experimentally. And then finally, we've got the transcript ID within which this variant falls in. So I just noticed that I actually had a filter applied to this whole table. So if you look above the table, there are buttons of different filters that you can apply. And if you did follow along in your own browser, you might have seen that it looks a bit different to you on yours, and that's because I had already filtered this table by missense variant. So I filtered this table to only show missense variants. Um, if you want to delete a filter, you can just click on this X sign here, and then you can see that the table just refreshed, and now it shows me all of the different variants within the gene. Just to show you how the filters work. So if you were to actually filter it by consequence, you can click this consequence filter button, and by default, all filters are turned on. So if you were only interested in, for example, frame shift variants, you can turn all of the filters off by clicking this left button. Turns it all off, and then you look for your consequence that you're interested in, and you just click that, and now you'll only see frame shift variants in the table. So don't forget to click apply to save your settings, and that just automatically updates the table. So now you will only see variants that are you'll only see frame shift variants. Also, if you have a particular variant you're interested in, you can just type it in here on the top right-hand side of the table. So you can filter by, for example, variant ID. And if you want to download your table, you can click this Excel icon on the right-hand side. And if you click it, so I've just done that, it's just updating. Okay, I was just going to show you how it looks like if you were to download the table. But basically, you can click it, and then you can choose different layouts that you want, how you want to download this table. Oh, there you go. So it just opens your system viewer, and then you can just save it on your local machine. I'm not going to save it, so I'm just going to click cancel here. Okay, so this is the variant table. Now I want to show you how you can view variants in the location tab. So here we looked at variants within the gene itself. And if we wanted to look at variants in the gene region, we want to go to the location tab. So that's just here on the top left-hand side. So I'll click that. And by default, it sends us to the region and detail view, which you can see here on the left-hand side. Just to recap, so the first view here on the right, we have the chromosome view. The second view, we have the 1 megabyte view. And then scrolling down, which is still loading, this will be the region and detail view. Okay, so it might take some time for this to load, and that's just because there's so much information that Ensembl is trying to show on the browser. So this is why you just need to be a bit patient with this view. Okay, so as you've seen with the demonstrations with Ben yesterday, we have this region and detail view with different tracks, so different annotations available. in this particular region that you've selected. So we can also add variation annotations to this view. And to do that, we're gonna scroll up to the top of this view, and there's this cog icon on the on the left-hand side here. If you click that, it opens a pop-up window, and this is just the this configure this page option. So this is where you can add the different tracks. On the left-hand side here, there are different categories. So we've got sequences of assembly, genes, and transcripts, which you've played around with yesterday. But then we also have variation data a bit further down. And if you click that, excuse me, if you click the first link in variation, you'll see that here on the right-hand side, you'll have a number of different tracks that you can turn on. So for example, you can turn on tracks of the 1000 Genomes project, which created a catalog of common human genetic variation data. So by default, there's already been some turned on. So here we've got, for example, all short variants turned on, and then also all common short variants. If you wanted to turn the track off, you can just click this box on the left-hand side of the title and just turn off. And then the same here. So here we have a particular population track turned on. So SAS stands for South Asian. If you're not sure what the track represents, or if you need a description of the track, there is this eye icon on the far right. And if you click that, it just gives you a little description of what data you can find in this track. If you click that again, it just closes that little window again. So I'm going to keep the South Asian tracks on, but then there are also other tracks that you can turn on. So there's different, for example, phenotype information, some more here, and then we've also got some copy number variant probes. So there are a lot of different tracks that you can turn on, which you can explore in your own time as well. So I'll scroll back to the top of the page. And once you're happy with your selections, so we've selected South Asian here, you can click this check icon on the top right-hand side to save and close. And this just updates our region and detail view. So you can see that it's saying here it's loading. Okay, so once it's loaded, you can find the tracks that you've selected further down. So in this case, we have it quite at the bottom of the diagram. And here we've got 1000 genomes, South Asian SNPs and indels, and 1000 genomes, South Asian common SNPs. So these are the tracks that we've just added. The different colored boxes or lines that you can see here represent the different variants that you can find. So let's say you're interested in this little green variant here. If you scroll down, there is a legend, which here, variant legend, and you can see that bright green is a synonymous variant. And these views are also interactive. So if you were to select that variant, you can click on it. And it's loading right now, but what you will see is a pop-up window, and it will give you more information on the particular variant that you just selected. So it'll have information like the ID of the variant, the consequence. Oh, there we go. So the ID of the variant, so in this case, it's this RSID. Then we've got the consequence, non-coding transcript, exon variant, and then also we've got the type of variant, so SNP in this case. You can close that, and you can explore all the other variants as well. So you can click around and see what information you can find. Then also, here we go. So by default, so we've turned these two on, but by default, we also have phenotype associated data turned on. So a track that shows phenotype associations. Again, the different colors or different lines that you see in this track represent different phenotypes. So if you scroll down, oh, where is it? Oh, I can't see it right now. But if you're not sure what, for example, the yellow means, you can click on it. And then you can see the type of this phenotype, and then also where or how the source that it's coming from. So let's close this. And now that you know how to view variants within a gene and also variants within a region, I want to show you how you can view a particular variant if you have one. So from our presentation, I have an RSID or a variant ID copied. So I will post that in the chat box. And we want to look at this variant in human. So if you want to do a new search, you can use this search bar on the top right-hand side, and I will paste the ID in there. And then you can also select this little Ensembl icon on the right, on the left, and just search human. And then search. Okay, so you might have noticed that we're now in a new tab. So we are now in the tab called Variant tab. And by default, you will land on the Explore this variant display. So here you have information about this particular variant. So for example, the consequence, the alleles. So we've got G, reference allele, and then A and C are the alternative alleles. We've got some minor allele frequency information, some different pathogenicity scores, location, also the different evidence or projects where this variant was identified and verified, sorry. Then we've got some information on clinical significance and also some synonyms. And then also the source from which it's coming from. So for this case, this was imported from DB SNP. Further down, you have some options available for you. So we've got genomic context, genes and regulation. You can view the flanking sequence and so on. But what I mainly wanted to point out was all these options, all these views are the same views as the ones here on the top left-hand side. So whether you click in one of these options or these options down here, you will land on the same page. So, for example, if you wanted to look at genes and regulation data, you can click this icon here, genes and regulation. And then you'll see on the left-hand side that you are indeed in this genes and regulation display. So you've landed on this display. If you scroll down, you'll have a table of all predicted consequences of this variant. So you might have noticed that a single variant, so for this case, it's RS4988235, and this variant can have more than one predicted consequence. And that's because a variant can be located on more than one gene transcript. So if you look at this transcript column, you'll see this is one transcript, this is the same, this is another transcript, that's the same. So there are, the variant can be located on multiple transcripts, and multiple transcripts describe, one gene can describe multiple transcripts. So we've also got the allele information, we've got the consequence type. So again, this is there's a dotted underline here. So that means you can view a description. Then you've got some more information on positions and also details if you want to see them. You can scroll down further and you'll see another table, which just gives you more information about gene expression correlations in different tissues. So you've got some stats here and then also the type of tissue that this variant was expressed in. So this is the genes and regulation view of this variant. You can also look at population genetics information of the variant. So if you scroll up to the top of the page, you'll see on the left-hand side a population genetics link. So we'll click that. I'm just going to scroll back to the top. So this is the population genetics view. And here you will find allele frequency information. So for example, you'll find information on the 1000 Genomes project. Then there are also other projects that have identified allele frequencies, so Nomad, NCBI Alpha, TopMed, and so on. Scrolling back to the top, just to look at the different frequencies in a little more detail. So for example, for the 1000 Genomes project, we've got these little pie charts, and they just give you an overview of the allele frequencies in each population. So we've got the total population, but then we also have these subpopulations, which are also divided into smaller populations. Sorry. So we've got African population here. You can hover over the name. And if you wanted to look at the subpopulations within the African population, you can click this link underneath that says subpopulations. And that just sends you straight to the subpopulation section for the African population. If you want to find out more details of particular populations, you can scroll down to the table. Again, we're still under 1000 Genomes project. And here we've got the African population again first. You can find out the actual numbers of the frequencies. So, for example, the allele frequency column, so the second column here, counts is the number of total allele. So as we humans are diploid organisms, we have two copies of each gene and therefore two alleles of a certain variant. So each individual that's been sequenced in this population provides two counts to the allele frequency. So the frequency, and then we've got the count in parentheses. Then we also have genotype information, genotype frequency information. So we've got the observed genotype of the individuals. You can also, if you click the show button on the right-hand side, you can find out a little more details on genotypes. So once you click that, you get sent straight to the bottom of the page. And here you'll just see genotype information of the individuals within the population. So for this case, this individual is male, has a GG genotype, and is within these populations. Scroll back to the top. Oops, that was a bit far. If you want, you can also export this table. So again, there is a Excel icon on the right, and then you can select which table you want to download. So whether you want to download the entire table or whether you applied a filter and just want to download whatever the filter is applied to. And then finally, I want to show you the phenotype data of the variants. So if you scroll back up to the top of the page, and we look here on the left-hand side, on the left-hand panel, there is a link that says phenotype data. And here you just you can find a list of all the different phenotypes that are associated with this variant. So you have the name, you've got the source, and then some accession numbers as well. There's publication links if there is publication available, and so on. So again, you can download this and save it on your computer. If you have a phenotype that you are interested in, you can also search Ensembl for your phenotype. So you can enter your phenotype in the search box, and you can do the search in a reverse order. So search for the phenotype and then look at the different variants that are associated with that phenotype. Okay, so that brings me to the end of my first demonstration. I'm just going back to the slides, and sorry, there. So now you'll have some time for the exercises. So just to go back to the training page, I've got a tab open here already. So if you go to training.ensembl.org, you can go to training materials here at the top of the page. And it will be the first link that you see. So St. Xavier's College. And here you'll find the variation exercises on the right-hand side. So if you click that, there are different exercises for you available. So we've got some human exercises, we've got some mouse exercises, some plant. So I'll give you 15 minutes to do those exercises, and then we will come back and I will talk you through the variant effect predictor. on Ensembl. Okay, hi everyone. I hope you had some time to try the variation exercises. Does anyone have any questions before I move on to the next module? All right. If you do have a question, you can always put them in the living document or in the chat box, and Ben and myself will get back to you. Okay, so let's move on to the next module, where I will briefly talk about the variant effect predictor, or VEP. So VEP is a tool that you can use to annotate and predict the different functional consequences of your own variants. So let's say you just received your sequencing data and you've called variants from this data, and now you want to find out more information. For example, the consequence, whether this variant has been identified before, and any linking studies. So this is what you can do with VEP. You can find out the effect of your SNPs, insertions, deletions, and structural variants. And this is how you can use VEP. So first, you input your data in one of these formats. So I'll show you what these formats look like, because I think it's a bit easier to understand what the formats are if I show you on screen. Then your data is processed by VEP and searches Ensembl and external databases to give you information, for example, the effect of the variant, the pathogenicity score, allele frequency, or other consequences. So there are three ways to use VEP. You can use the web interface in the Ensembl browser. You can access VEP through the command line or REST API. But today I will show you how to use the web interface. So in this demonstration, we will look at a set of 5 variants on the human chromosome 9, and we are going to find out if the variants have already been annotated in Ensembl, what genes are affected, and whether these variants affect gene regulation. So for this, we will change to the Ensembl tab. So I have one open still from the first demonstration. I will just move to that. And to go to VEP, all you need to do is click this VEP link at the top of the page, so at the navigation bar. So I'll click that. And here you'll see the different interfaces of VEP. So web, command line, information on how to access it through the command line, and also REST API. But we are interested in the web interface, so I will click this big button that says Launch VEP. And I will delete. So this, at the moment, you're seeing jobs that I've done in the past. So I've done one job here in VEP. I'm going to delete that so we can see the same things on our screens. Just delete that. Okay. So this is what you should see if you haven't done any queries using VEP. So we've got this first section, so the species you're interested in. In our case, we're interested in humans, so we will keep Homo sapiens here. But you can also change the species by clicking this Change Species link. And that opens a pop-up window where you can either search for your species by typing it into the search bar, or you can just search for it using these different categories. So we've got human selected already, and you can make sure that you have indeed selected it by looking here on the right-hand side. And then don't forget to save and close. So I click apply here at the bottom of the page. And that's our species. So then next, we can give this query or this job a name. So let's just call it web demo. And then next, this is here under input data. This is where you paste your variants. So I mentioned during the presentation that there are different formats that you can use. If you look here under the box where you paste your data, there are links for different examples. So we've got the Ensembl default format. So if you click that, you can see that you just have a short list of variants in the Ensembl default format. Then we've also got the VCF format, variant identifiers, so RSIDs, HGVS notations, and then also in the speedy format. So from our demonstration slide, we haven't formatted the variants in the correct way yet. So you'd have to do that first, but I've already got it copied here actually. So I'm going to paste the formatted list of variants into the chat box. And this will be the Ensembl default format. So now that you have it on the chat box, I'll also paste it into our web input data. Just remove the empty rows. You don't have to do that, but I like to make it look nice. So you've inputted your list of variants, and then, oh yes, so if you have an even longer list or even a file, you can choose to upload the file instead of pasting it. Or if you have a URL to your file, you can also paste that here under provide the file URL. Next, we need to choose which transcript database to use. So if you hover over it, it just gives you a bit more information of the different databases. So we've got GenCode, BASIC, and RefSeq. We will stick with the Ensembl GenCode transcripts, but you can also select one of the other, the RefSeq database, or you can also choose both databases, so both Ensembl and RefSeq. Next, we have options to apply additional configurations. So in the Identifiers tab, the first one here, if you click that, you'll see different options that you can turn on. So you can choose to display different identifiers within which the variant falls in. By default, the gene symbol and transcript version identifiers are turned on, but you can also turn on other identifiers like protein or UniProt IDs. Then if you close the identifiers tab and open the next one, variance and frequency data, you can choose here whether your input variants are known or novel. So this is where you can find out whether a variant in your list has been identified before or not. And this is turned on by default. So yes, I want to find out if the variant has previously been identified. Then also you can turn on the option to show PubMed IDs for citations. So if you do have a variant that had been identified before, you can get a link to the actual publication. So let's close this tab and open the next one. So here we can select additional annotations. So you can, for example, turn on annotations about the transcript biotype, or a bit further down, you can find out if the variant falls within a regulatory region, which is turned on by default by looking here on the right. And then you can also look at, you can also turn on phenotype data and information and citations as well. So let's close this additional annotations tab. And then you've also got prediction, pathogenicity prediction scores under predictions. So these are the different scores that I've shown you in the first presentation. And by default, we've got SIFT and Polyphen turned on, but you can also turn on other scoring systems. And then finally, I want to show you the filtering options. So by default, we don't apply any filtering to the to your list of variants, and but you can choose to turn some filters on. However, just note that if you do apply a filter before the actual job, you might lose on potentially useful data. So it's good practice to apply filters after you've done the job. So once you have your results, you can also apply filters then. So by default, we have this turned off. And then once you're happy with your settings, you can just click Run here on this green button. And this sends you to the display where you have all your jobs, all your web jobs listed. So because I deleted the first job, I only have this one job here. And you can see it's still queued, so it's still working away. And this page refreshes every 10 seconds. So you can see here how many seconds are left. You can also refresh it in your own whenever you want. Then we've got on the right-hand side here, you have different options for your job. So if you have an account on Ensembl, so you can register here on the top right-hand side. If you have an account on Ensembl, you can save your jobs and look at them later on by clicking the Save button icon here. Then you can also edit your query. You can share the URL to your query. So if you want to show your collaborators what you found, you can click this button and then share the share this link with them, and they will be able to see your results. And then finally, you can delete your job here. So once the job is done, you'll see a button up here that says done. So you can click either on that green button or view results to see your results. And here you'll see your web results. So we've got some summary statistics first. So for example, here we have the number of variants processed, then the number of variants filtered out. Because we didn't apply any filters, no variants were filtered. And then we've got the number of how many of your variants have been previously identified and how many of them are novel. So one of the variants is novel and the other four have previously been cited. Then we've got the number of overlapping genes. So it's overlapping 4 genes and 24 transcripts. And also it's overlapping, our variants are overlapping 2 regulatory features. Next, we have a pie chart of the different consequences of this of the variants that we've inputted. So we've just got some percentages, and then also the coding consequences. So whether it's a synonymous or non-synonymous or missense variant. Then scrolling down, you'll see your actual results. So first, we have the name of the variant. So we called our variants var1, 2, 3, 4, 5. So if you want to sort the table by the variant name, you can click uploaded variant, and that just sorts it by the name. Then we've got the location, the position information, the allele consequence. So for this first one, we've got a consequence of missense, then the gene names, the transcript name ID, sorry, what type of transcript it is. So in this case, the first one is protein coding. We've got some more position informations. And then also here under this existing variant column, you'll see if there is a link available, it means that the variant has previously been identified. Whereas if you scroll down, there are also entries without a link. So this means that this variant is novel or it hasn't been cited before. And then scrolling further down the table, you've got the pathogenicity scores. You've got a link to publications and some other regulation information. So if it falls within a transcription binding site or motifs and so on. So if we go back to the beginning of the table, you can see that there are multiple entries per variant. And this is because a single variant can have different consequences in the different transcripts of a gene. And we know that one gene can be transcribed into multiple transcripts. So that's why you have so many entries for example, the first variant only. You can also filter this table. So if you go to the top of the table, just above it, you have the section that says filters. And here you can select your, in this drop-down menu, you can select your column that you want to filter. So for example, if we wanted to filter the table by missense only, we can select consequence, is, and then whatever consequence you're interested in. So let's say missense, and then just select it from this drop down here. And don't forget to add, because otherwise Ensembl won't apply these filters to the table. It's just taking a little time to load. If your internet was ever a bit slow, you can use one of the Ensembl mirrors. So I'll just show you how to go to them after I finish this demonstration. Okay, so we just applied a filter, so you can see here consequence is missense variant. And now you can see in the table, you'll see that we only have variants that have a consequence of missense now. You can apply multiple filters as you wish, or you can also edit your filter or delete or remove the filter by clicking this X here. If you want to download your web results, you can select one of the different formats that you want. So depending on your downstream processing, you might need to select a VCF format or just a plain text format. And yeah, I think that's everything I wanted to show you. So I've shown you how to export it, how to apply the filter, how to close, how to remove a filter. Yes, I'll just show you how to find our mirror sites. So if you notice that Ensembl is being very slow for you, you can visit one of our mirror sites by going to Help and Docs here at the top of the page. So I'm going to open that in a new tab. And here on the left, you can just search for mirror sites. So it's a bit further down, and click the link. And for example, if Ensembl would have been super slow today, I would have used one of these mirrors just so that Ensembl is runs a bit faster for you. So you can click on them, and you'll know you're on a mirror site by looking at this little icon next to Ensembl at the top left-hand side. Okay, so that was the demonstration for the VEP. You'll have some time to do the exercises now, so I'll give you, I think, 10 minutes to do the exercises, and then we'll come back and I'll talk you through the regulation data that we have available in Ensembl. Hi everyone. I hope you had a quick break. Does anyone have any questions before I move on to the next module? Okay, so if not, I'll move on. Let's talk about regulation data on Ensembl. So in this module, I will give you a brief introduction to the regulation of gene expression and talk about the Ensembl regulatory build, so how regulatory data is generated and where this data comes from. On Ensembl, a number of genomes have been annotated with regulatory elements or features that control gene expression. For example, promoters, enhancers, and repressors. To annotate these regulatory elements, we look for specific biological data types, for example, epigenetic marks, so histone modifications and DNA methylation, transcription factor and polymerase binding patterns or sites, or by looking at open and closed chromatin conformations, which is by looking at DNase 1 sensitivity. Essentially, each cell of an individual shares the same genome and therefore the same genes. But we also know that there are hundreds of different cell types and very distinct phenotypes. And this is because of different expression levels of different genes. That's why the regulation of gene expression is so important. So let's look at the different blood cell types, for example. They all have very distinct functions. For example, red blood cells are important in oxygen transport, or basophils, a type of white blood cell, are involved in allergic reactions. All of these cell types are derived from a single hematopoietic progenitor cell. And what makes blood cell types different is how they express genes at different levels and at different times. So we can say that these different cells have different epigenomes or different epigenetic expression profiles. You can think of transcriptional regulation as layers of controlling the different timings and expression of genes, which you can see in this diagram. So we have a strand of DNA, and here at the bottom left is the coding region of your gene of interest. Upstream of the gene, you can find a promoter region. Excuse me. You can find a promoter region which binds RNA polymerase, the enzyme that transcribes DNA into mRNA. And the promoter region also binds transcription factor proteins. These are sites, also known as transcription factor binding motifs or transcription factor binding sites. And transcription factors are involved in inhibiting or stimulating the transcription of a gene. Another regulatory element are enhancers. So you can see that here. These sites are often located a bit further away from the coding region of the gene, and enhancers have their own set of transcription factor motifs, and they bind activator proteins, which promote the activation of transcription in the promoter region. And then similarly, we also have repressors and insulators that bind specific proteins to downregulate gene expression. So the binding of proteins to these transcription factor binding motifs is regulated by methylation. Within the DNA sequence, there are regions known as CPG islands, and these are regions that are rich in CG repeats. An enzyme called DNA methyltransferase moves along the genome and modifies the C bases within these CPG islands with methyl groups. And this gives different methylation patterns, and these methylation patterns can modulate the binding of transcription factors. So for example, methylation can block a transcription factor from binding to a transcription factor binding motif. So the transcription factor sites. So DNA methylation is one layer of transcriptional regulation, but there are many other layers of transcriptional regulation. So, for example, the conformation of the chromatin can direct the activation or deactivation of gene expression. So in this diagram, you can see that the chromatin is usually bound in a uniform manner. The DNA strand is wound around 4 histone proteins, making structures called nucleosomes. So we know that there are regions of chromatin densely packed together, and that is associated with transcriptional deactivation. And we also get regions of chromatin that are more loosely packed. And this is associated with transcriptional activation because the DNA is more accessible to transcription proteins, so they can access the DNA strands better. We also know that the position of the nucleosome, so these structures here, and the modification of the histone tails, so which you can see depicted in this blue line here. The position of the nucleosome and the modification of the histone tails themselves, they can affect transcriptional regulation. And then finally, the actual 3D conformation can also regulate gene expression. These layers of transcriptional regulation can be assessed by different experiments by looking at the expression patterns of a gene. So this diagram just shows you the different techniques that can be used to determine these layers of transcriptional regulation. For example, we can use DNA sequencing to assess open or closed regions of chromatin by looking at the sensitivity to the DNase enzyme. We can use ChIP-seq to understand protein binding patterns and histone modifications. And we can also determine gene expression levels by looking at RNA-seq data. So now that I've given you a brief introduction to transcriptional regulation, I'll talk about the process of how Ensembl uses this kind of data to annotate regulatory elements in the genome. So first we take these different types of data and import them from different sources. Then we process the data to predict the positions of regulatory features, so the promoters, enhancers, transcription factor binding motifs, and so on. And then in step 3, we assess the activity of these different regulatory features in different cell types. And then finally, we display the regulation data on the browser. So where does the regulation data come from? There are 3 main projects that Ensembl takes data from. One is the ENCODE project, which looked at 48 different human and 8 different mouse cell lines. They generated data that looked at histone modifications, transcription factor binding motifs and sites, RNA polymerase activity, and DNase sensitivity. The Roadmap Epigenomics project looked at gene expression in cultured primary cells. And then finally, the Blueprint Epigenome project generated ChIP-seq and DNA sensitivity data of 20 primary cells. On Ensembl, we only have regulation data for human and mouse, but we are working on expanding this to other species in the future as well. So in the first step of the Ensembl regulatory build, we plot the raw data against the genome using several different algorithms to annotate peaks. So you can see the raw data is colored in these squiggly lines, and then the reference genome is represented by this straight blue line here at the bottom. So for example, we start with data that assess the activity of transcription factors and histone modifications. So here, transcription factors B and C and histone modifications 1, 2, 3 display high activity in this particular region of the genome. When the activity hits a certain threshold, and this is where the peak gets annotated. And they are we say that they are all co-located in this region. And this is important to know for the next step. So in this step, we move along the genome and find out if any of the other regions have that exact same pattern of activity or co-located regulatory elements. If any of those positions are, for example, known promoters, we infer that all the other positions with the same pattern or signature are also promoters. So we predict these feature positions. Then we look for other patterns and we do the same process again. Um, Next, we categorize the different regulatory features based on the underlying pattern or signature. And this process we call segmentation. So for example, this particular pattern, oops, I'm so sorry about my mouse. There you go. So this particular pattern is known to be, for example, associated with a promoter sequence, or this pattern here may be known to be associated with an enhancer. In step 3, we predict the cell type activity. And we do this for every cell line that we take from the different project sources. So here we move along the genome in 200 base pair blocks, and we ask, is there a regulatory element annotated in this particular block? If there is, we annotate that block. So this track here on top that you can see is what we call our multi-cell regulatory build. It's the annotation of regulatory features for all cell types combined. Next, we take this multi-cell regulatory build. We look at the individual cell types, and we check the activity of the regulatory elements. So we ask, is this element active, inactive, poised, or repressed? And to determine this, we look at the different experimental evidence provided in the project sources that I mentioned earlier. And then finally, we display this information as tracks on the Ensembl browser. I want to highlight that Ensembl does not link regulatory features to genes. So we simply provide you with the locations of the features. We also don't link regulatory features to gene expression, but rather we present cell line specific regulation data and tissue specific expression data. So therefore, when you are using Ensembl, we do not make inferences about the regulatory data, and it's up to you to make your own inferences. So for this demo, we are going to look at the KPNA2 gene region. We're going to find regulatory features and then explore its expression in different cell types. So as usual, I'm going to copy the gene name and paste it in the chat box. And then I will go to the Ensembl tab. So I still have one open here. But if you want, you can just open a new tab. So we are interested in the KPNA2 gene in human. So just like before, I'm going to use the search box here on the top right-hand side and paste the gene name. And then we've still got human selected here on the right. So I'm going to keep it that way and click search on the right. Okay, so we're interested in KPNA2 in human. So it's the first hit. I will click that link. And that leads us, lands us on the gene tab, which you can see here on top. But for the demonstration, I actually want to show you the location tab. So how to view regulatory features in a region rather than in a single gene. So for this, I would click the location tab on the left-hand side, where we land on the region in detail, and we'll scroll down to the most detailed view, which is still loading. Okay, so we have different ways of presenting regulation data on Ensembl. So if I actually go back up to the 1 megabyte view, you can see here. So we've got our gene of interest, KPNA2, highlighted in green here. But you can also see there is a track that's called regulatory build. So this is just an overview of the different regulatory features that you can see in this region of the genome. And the different colors represent different features. So if you look a bit further down the image, you'll see this regulation legend, and you'll see the different colors representing different elements. But if you wanted to look at a region in more detail, you'll want to scroll down to the region in detail view. And here, if you scroll to the bottom of the page, you'll also have this regulatory build track. But this is because we are zoomed in quite a bit in this diagram. We can only see, in this case, I'm just scrolling down to the legend. So here you can only see the promoter region, so dark red, and then also the promoter flanking region in light red. You can, so these are interactive. So you can click on the different regulatory elements. So if you click on the promoter, you'll find more information such as the stable ID, the type. So we know it's a promoter because we just looked at the legend, but then also just the exact location. So if you want to find out more about the promoter itself, you can click on the stable ID, which I'm going to do now. And that just opens a new tab here on top, the regulation tab. So regulatory features on Ensembl have their own Ensembl ID. So for human, in this case, it starts with ENS and then R for regulatory feature. Okay, so we are on the regulation tab now, and this just shows you an overview of the regulatory feature that you just selected. So we just selected the promoter feature, the promoter region, and here you can see it in just a little more detail. So we've got here under regulatory build track, we've got the flanking region of the promoter. Oh, we also got the promoter region itself. If you're unsure about the colors, there is a legend at the bottom of the diagram as well. So we've got our regulatory track, but then we also have a number of other tracks. So we've got the Phantom 5 track here, which is the functional annotation of functional genome track. So this was a project which looked at annotating the functional regions in the genome. You can also find more information by clicking on it, and there is a link that leads you to the project site as well. Then we also got a track, a char-based track, which just gives you microRNA targets. And then this last track is the motif features track. So these are the transcription factor binding motifs or transcription factor binding sites. This is where the transcription factor proteins can bind to. So we have gray and then also black blocks or lines that you can see here annotated. The black lines represent experimentally verified regions, sites, and the gray blocks are predicted sites. So if you click, for example, on a black box here, again, it's interactive. You can find out more information um, about the different um, experiments that this was verified in. So, for example, here it was verified that the transcription factors TEAD4 and ERG both bind to the site, and this was experimentally verified in this list of cell lines. So I'm just going to close this pop-up window, and I will scroll down to the bottom of the page, where we have a table of a summary of regulatory activity in different cell lines. So here we can see in which cell lines this regulatory feature, so this promoter, is either active, inactive, poised, or repressed. And you can look at each of these cell lines in more detail by scrolling up to the top of the page. Sorry, there's a lot of scrolling in this involved, but once you get used to the structure of Ensembl, um, it will make sense. So we just looked at the table of cell lines, the activity in cell lines, but we can also look at the individual cell line activity. So you can do that by clicking details by cell type. So this icon here. And you can see on the left-hand side, you can see that we are under the details by cell type display. It's just loading right now. But what you will see here is you can, oh, there it is. So here you can add tracks to show the activity of the different cell lines that you're interested in, and then also the corresponding evidence. So to add a cell line, you just have to click Configure Cell Tissue at the top of the diagram. And here you'll see, just like before, the different categories of tracks that you can add. But right now we are under regulation here on the left, under regulation in features by cell and tissue. So first you'll have to select the track, the cell type that you want. So we'll click on this number 1 icon here on the right-hand side. And here you can either search for cells or tissues by typing in your name of interest, or you can also just go through the alphabet and just search for them here further down. So let's say, for example, if you're interested in HeLa cell, in the HeLa cell line, you can select that here. So at the moment it's green for me, that means it's already selected for me. And then let's also just select another one, A549 cell line. So again, you can click that, green means it's been added, and then you can make sure you can check the different cells that you've selected here on the right-hand side. So we've got 4 selected out of 148 available. And if you want to see which ones these are, you can just click on this plus icon on the right. Excuse me. And here, again, you can see a list of the cell lines that you were interested in. You can also turn them off here, so you don't have to go through the whole list. Next, you can click on this Experiments tab here on the top. And here you can select the different experimental evidence that is available for each cell line. So it's split into three categories: histone activity, histone modifications, open chromatin conformation, and then also transcription factors. So there are different experiments that you can do to assess histone modifications. So these are just a list of these experiments. Then also for chromatin, there's only one experiment that we extract data from, so DNase I sensitivity. And then also transcription factor experiments. So here again, you can either search for it in the search bar on top, or go through the alphabet and search for your transcription factor of interest manually. Once you are happy with your experimental evidence selection, you can click this green button at the bottom right of the page that says Configure Track Display, and that just saves your settings. But now you can see we are under Configure Track Display here on the left, on the top of the page. And here you can choose how you want to view your data on the track. So you can select the format of how you want to see the data. So let's say you want to look at this particular cell line, A549. You can click this little box, and here you can just choose to show the column, the experiment, so the columns, and then also the cell line. So if you turn that off, it means that you are not showing it on your track. You can also do that individually, but for the other cell lines as well. So if you have the different views turned on, the different options turned on, you can also choose the way you want to display this information. So you can show it either as a peak or as a signal, so like a squiggly line, or you can show it as both. So you can play around with the different types of views that you want to see, that you want to show. Okay, so if you're happy with how you want to show your data, so I've just kept everything in the default setting, you just need to click View Tracks to save your settings. And this just automatically closes the pop-up window and updates our view here. It's just taking a little bit of time. But while that's loading, I can show you what we already have in the view. I hope you can see it. So we have our usual region and detail tracks. So we've got the context track, which is the sequence track, our regulatory build track. So because we earlier we selected the promoter region, we can still see this here. Then next we also have so we have the first cell line that we've selected. So in this case, it's the astrocyte tissue specific cell line. And you can see the different, so the sites. Um, okay, no, hang on. So we selected to show different experimental evidences of the sites within the astrocyte cell line in this promoter region. And here you can just see this in a graphical way. So for example, um, I won't be able to click it right now because it's still loading. But if you click the different features, you will see um, more details about what was found in the experiment. So the expression of of the promoter in the astrocyte tissue. Um, what I might actually do is I will open a new tab and use one of the Ensembl mirrors because I want to show you just a little more detail, the different tracks, and I won't be able to select any by staying here. So I'll just open a new tab on my browser and I'll go to asia.ensembl.org. Or I'll use one of the others. US West is also another one, uswest.ensembl.org. Okay, so the Asian mirror is a bit slow, so I'll close that tab. And then the usual server is also quite slow, so I'll leave it open, but we'll stay with the Ensembl US West mirror for now. So I'll just search for this regulatory feature that we were just looking at, so the promoter. Instead of going through searching for the gene, I'm just going to search for the regulatory feature straight away. So it's this one, human regulatory feature. I'll click the first link. Then it's updating here, but we were interested in looking at particular cell lines, so I'll just click that icon, details by cell type. And then because we haven't selected anything for the within the US West mirror, I'll just need to add those tracks again. So click the configured cell tissue button. Then we'll just select a number of different tissues or cell lines, astrocyte. Look for some HeLa cells. There we go. And then make sure that the experiments are all selected. So I'm going to turn all histone modification experimental evidence on. Then also for chromatin, open chromatin, and then the transcription factors as well. So here on the right, we can see we've selected 3 cells or tissues, and then the different experimental evidence. Then I'll click Configure Track Display. And we will leave all of this as it is, so the default setting. And then we'll click View Tracks again. Just go back to the first tab. Okay, so right now we're seeing an error, and this means there's something's gone wrong on our end. If you ever see an error like this, you can actually email us on helpdesk at ensembl.org, and we will look into it and tell our colleagues that to make them aware that there is an error so that they can check what's going on. And this is taking a long time now as well. And I think this is the case because of that error that's going on. So I won't be able to show you more details of the different regulatory feature tracks, but in your own time, you can uh, click the different features and just read more about them and just get familiar with the view. Um, so yeah. Um, okay, well, you might be lucky. It might work for you. So if you want, you can try the exercises under the regulation tab in the training page. Um, and then we'll be here if you have any questions. Oh, actually, we have screenshots actually of these walkthroughs. So if you go to the training page, so this is the training page for St. Xavier's College, and then we've got regulation, the regulation exercises here on the right. Um, so there's a section, the regulation demo section. And here you'll just be able to see the different steps that I've just gone through in the demonstration. But what you can also do is, if you scroll down, so here it just shows you what um, how to add the different displays, the different formats of displays in the tracks, and this is the view that I originally wanted to show you. So it's not loading right now, but here, so we started off with the sequence track. We have our promoter track here, but then when you have your cell line selected, so this is the first cell line that we've selected. In this case, it's aorta. You can see the different features are annotated. So the different sites where this particular experiment um, had a result. So where the transcription factor um, was expressed. Sorry. Um, okay, so to do the exercise, to do the exercises, you can scroll down to the bottom, and then you can just go to regulation exercises. So you might not be able to do the exercises right now, but please feel free to do it in your own time, or we might actually have time tomorrow after at the very end, where you can just go through the regulation exercises yourself, and then we can also discuss if you have any questions. Let's see. Wonderful. Okay, so I think it might have just been my internet, but it's loaded now. So what I wanted to show you originally is to click on one of the experiments tracks. So you can click on the different features, and you can just see here. So it opens a little pop-up, and you can just see here, for example, the project that this was done in. So in this case, it's encode, and then you can click the others as well, and then again encode, also the name of the feature, and then also the position. So feel free to get to know this view and click the different features and learn more about them. So I'll go back to a presentation, and I'll give you, let's say, 15 minutes time to do the exercises. Hopefully it works for you. And we'll be here for questions if you have any. Hi, everyone. So it's 4:50 now in your standard time. I hope you had some chance to try the exercises. Does anyone have any questions before we wrap up for today? Okay. Well, thanks, guys, for today. I really enjoyed presenting the modules today. And tomorrow we will look at comparative genomics data on Ensemble. Then I'll show you how to use Biomart. And then finally, Ben will talk to you about how you can visualize your own data um, on Ensemble. But for today, we are done, and we will see you again tomorrow at 2 o'clock India Standard Time, the same link as today. Thanks, everyone.
Thank you, everyone. Thank you, Luis. So we will meet on tomorrow, same time, 2 to 5 IST, and the same link.
That's right, yes.
Thank you,
Yeah.
Luis. Thank you,
All
Ben.
right.
Thank you, ma'am.
Thank you, everyone.
Yeah.
See you tomorrow.
Thank you for this wonderful presentation.
Thank you. Bye.
Bye bye.